I'd like to give a demonstration of the IDR search engine, and I'm going to start at the home page of the IDR at idr.openmicroscopy.org. So this page in the lower half here gives a nice overview of the type of data in IDR. This is grouped by the different studies, the 141 different studies in IDR, and by mousing over these I can see the number of images within each study, and I can browse to see these images if I see something interesting. But if I want to search for something specific among those 14 million images, I can use the search engine. And I have the option here to search by attributes that are on the studies themselves, such as title, organism, or author. But I can also search for attributes that are on particular images. And this is just a small selection here. So I'm usually better off by searching directly up here for attributes that will cover all available attributes. So I'll just start with something quite generic to illustrate this, and in the top half of the search autocomplete results I'm seeing attributes that are matching various different studies in terms of imaging methods and descriptions, and I'll also see authors. And in the lower half here, these are matching attributes on images. Let's search for a particular gene, and again I'm getting some matches for OR in the studies, but also a number of attributes on images. And if I'm just interested in this exact match here, I can just hit enter to perform that search. So the search engine is returning results for gene symbol equals OR, and it's found 3 studies that contain matching images. I can modify the search up here by choosing contains, and that will give me a much larger number of results. And I can also perform a new search up here. Again, if I type in OR, I can see the various other genes that start with AUR, as well as a number of other attributes. And these are sorted because I have gene symbol here to show gene symbol first, but it covers all attributes to be sure that I don't miss anything. Let's search for different gene, and I'll just hit enter to perform that search. And so here I'm getting all the different studies that contain matching images, and if I want to view the images within any of these studies, I can just expand. And now we're performing another search from the search engine to show images that match that criteria but are also coming from that particular study. I can use the I link here to browse to those images within the plate, or I can open an image viewer, but simply by clicking each of these images I can upload a lot more details. So these are all the attributes on those images that are being indexed by the search engine. So this is an overexpression cell painting study, and so we have a number of different phenotypes that are coming from the overexpression of KRAS, and we have here the phenotype as generated by the author and submitted to IDR, but as part of the curation process, IDR has mapped these terms onto phenotype term names. And by using these ontologies, we're more easily able to compare images across the various different studies submitted to IDR. And I can perform a search for these just by clicking on any of these links in the right hand panel here. And this has performed another search, phenotype term name equals decreased cell size phenotype. And again, we have all the studies that have images with those phenotypes. So again, I can browse the images within that study. I also have a link here to take me to the home page for that particular study, and this will give me information about the study, links to the publication, the type of screen for this images, and a recent addition to this page, we have here a tool called the BioFile Finder. This allows us to perform additional searches for within these images. So I can load all the images for that study into the BioFile Finder, and these are the 41,000 images as indexed by the search engine, and the search engine is providing a table, in this case a Parquet file, that will be loaded into the BioFile Finder. Okay, so I have all the images within that study, 41,000 images. I can view the attributes for individual ones, and then I can perform a number of filtering or grouping options up here. So let's group by gene symbol. And so now I can see for each individual genes the images where that gene was overexpressed. And then I can do additional grouping options here. So let's group by phenotype term name. And now for each gene, I can see the various phenotypes that arise from overexpression of that gene. When I click on an image, I can use this link up here to open that image in the IDR image viewer. Okay, thanks for watching.